Hello students, welcome back to my biology online classes. Today I am here for delivering a lecture about another types of RNA. That one is tRNA or transfer RNA. In my previous lectures I have already discussed with you about in detail structure of mRNA. So today I am going to discuss another types of RNA, which one is known as tRNA and transfer RNA. Transfer RNA or tRNA, as the name suggests, it helps to transfer amino acid. So let's have a look about all the detail of tRNA and its structure. tRNA is a small, well characterized RNA molecule mainly which have a great role in protein synthesis. Transfer RNA is also known as adapter RNA. Concept of an adapter to provide the interface between nucleic acid language and protein language was introduced by Watson and Crick in 1955. RNA also participate in non-protein synthetic processes such as a primer during reverse transcription in retrovirus life cycle. So let's write the characters of tRNA. Point number 1. Like all RNA, tRNA is also single-stranded and the direction will always be 5' to 3'. Next point, tRNA is single-stranded but due to too much folding it seems to be double-stranded. This one is smallest form of RNA, normally contain 75 to 95 nucleotides. Next point, all of we know that we have in our body 20 essential amino acids. Which are called essential amino acids? Essential amino acids are those which cannot be synthesized in our body spontaneously, but they should be supplied through diet from exterior source as diet source acids. So there are 20 amino acids, and for these 20 amino acids 20 tRNAs are required because each amino acid carried by a specific tRNA of that amino acid. So let's have a look. For each 20 amino acids, 20 types of tRNA are formed, and each amino acid is carried by each 20 types of tRNA, carried by 20 types of tRNA. The next point, molecular weight of tRNA is 25,000 to 40,000 Dalton. Point number 6, this one is also called soluble RNA. Soluble RNA, and it is soluble in 1 molar sodium chloride solution. Solubility of tRNA, that it is soluble in which solution? 1 molar NaCl solution, sodium chloride solution. Next point, it is also called adapter RNA. Adapter between which? Adapter between nucleic acid language of DNA and protein language. It carries activated amino acid to the site of protein synthesis. The next point, the best model to study the structure of that one was given by scientist Robert Hole and their colleagues in 1965. Robert Hole in 1965, and the name was clover leaf model. This one is a very common term, we are very used to this term, the clover leaf model. So why it is called clover leaf model? Leaf-like structure. It has a leaf-like structure. Like what? Clover. Clover, what is clover? Clover means this one is a algae and it has trifolium structure. Clover or trefoil. All these clover and trefoil are names of the common genus Trifolium, flowering plants in legume or Fabaceae family. Okay, this one is from botany lecture. Just it's a curiosity and answer of this curiosity why it is so named clover leaf model. So this clover leaf model, this one is like this. Now let's draw the tRNA structure and it will be clear to you that why it is called that clover leaf model. Now let's have a look about tRNA structure given by scientist Robert Hole in 1965. Now let's draw the tRNA structure. Not very simple or not so complicated. So this is clover leaf model. Clover leaf model. Okay. Look this structure. This is called stem. Stem structure. Okay. Stem and loop. Stem loop structure. It's basically clover leaf model by of tRNA is basically stem loop structure. These are called stem. Stem, also called stem and stem, and loop structure. How stem structure is formed and how this loop structure is formed. Stem structure is formed because in this both sides there are complementary bases. We all know about the complementarity. That one is A. In case of DNA, A makes double bond, double hydrogen bond with T. But in case of RNA, we already know that we already discussed in my RNA polynucleotide structure that there is absence of T in case of RNA. Uracil is present in case of RNA. So adenine makes bond with uracil and G makes bond with C as triple hydrogen bond. As usual in case of DNA, the GC bond was the same type, but A makes bond with uracil, not with thymine. Although tRNA, all RNA, they are single stranded, but when these two complementary complementary bases are come in close contact with each other, they can form temporarily bond and looks like a stem stranded double stranded structure, but really it is not stem. And in loop, why in case of loop form, in this loop region there are presence of some unusual bases which cannot find its pair for pairing with another bases, so they are forming loop structure. So here in this case, here the loop, here the loop, and here the loop. In this 3 loop region of tRNA, there is presence of abnormal bases which cannot be paired with any other bases present normally in RNA structure. So they cannot pair, they cannot form stem structure. They are here singly, okay, and forms loop structure like here, here, and here. And which are these abnormal bases? Surely I am going to discuss with you in this cloverleaf structure of tRNA. So let's discuss about this structure in detail. tRNA is formed 5' to 3' end. So this one is 5' end, and this 5' end there is a G. 5' end is open here with G, and if G is present, so here parallel to G, C is present here. C is present here, as G and C are complementary in nature. And look at this region here, CCA sequence is present here, and this one is 3' end, and here OH, hydroxyl group is freed here. So this is the signature sequence of tRNA molecule. Okay, here 3' end, here at 3' end, OH, hydroxyl group is free here, and there is a sequence of CCA at 3' end and 5' end. Rather than 5' end, this one is the main signature sequence of identifying sequence of tRNA molecule, that is CCA structure of 3' end, and at the 3' end OH hydroxyl group is free. Okay, and this one is known as activated amino acid attachment site. This 5' and 3', this one is known as activated amino acid attachment site, and this activated amino acid attachment site contains 7 base pairs. 7 base pairs. 1, 2, 3, 4, 5, 6, 7 base pairs. Okay, 7 base pairs and CCA sequences. Variant base is present. Variant base is present. So here 7 base pairs and 4 nucleotides present here at activated amino acid attachment site. This one is known as first arm. Okay, this one is second arm. Here 2, second arm. This one is. Here 5 base pairs are present. 5 base pairs and 7 unpaired nitrogen bases. 1, 2, 3, 4, 5, 6 and 7. 7 unpaired nitrogen bases. Here as unpaired bases T is present. As all of we know that in case of RNA T will not be present, but here this T is present, and T here thymine is present, and this thymine is known as ribothymin. This T is known as ribothymin. It is a special base. Okay, here psi. It is a psi. This is a Greek word. Psi is 23rd letter of Greek alphabet. 23rd letter of Greek is also known as pseudo uracil, not true uracil but pseudo uracil, and it is called as psi. Okay, and it is responsible, this pseudo uracil, this is responsible for this loop formation. So it cannot find any complementary bases with which it can pair with, so it forms a loop structure. This one is known as variable arm. Here the presence of nucleotide varies. It has not a fixed number of nucleotide bases present here, so it is known as variable arm. After this variable arm, this one is known as anticodon arm. Here third arm. This one is known as anticodon arm or third arm. Anticodon arm or third arm, and here in anticodon arm 5 base pairs are present. 5 base pairs are present. So is anticodon arm 5 base pairs plus 7 unpaired nitrogen bases is present here. The abnormal base present here is inosine. And also like this pseudo-uracil, it also can't find its complementary base, so cannot able to form a stem structure. So for this inosine, here is a loop structure, and here this one is, here 3 codon is present, and say for example if mRNA is here, and mRNA carries AUG for methionine initiation codon, what base will be present here? So with A, all of we know A makes bond with T, but in case of RNA it makes bond with U. So here U will be present, and here U, so here A will be present, and here G, complementary base C will be present. So these have, and these anticodon arm of this tRNA have anticodon, which anticodon means codon present in mRNA and anticode is present here. So here in this in the case of this type of RNA, this type of RNA is code for, it has anticode for methionine amino acid here start codon. Okay, is it very clear? This is anticodon arm, and now let's come about the fourth arm. This one is fourth arm. It is also called D-loop. Why it is so named, I am going to discuss here. 3 to 4 base pairs. It has 3 to 4 base pairs along with 7 to 11 nitrogen bases, which are unpaired. So surely in this loop structure also a presence of abnormal, not usual nitrogen base is present. Surely as per knowledge we have already, there is also presence of a special type of carbon base. Uncommon bases are responsible for formation of this loop structure. So there should be a single uncommon hydro, D for dihydrouridine. Okay, here DHU, and this one is abnormal base. And this arm is also known as amino acyl synthetase binding site. Here amino acyl synthetase binding site. Okay, and this one is fourth arm. So this is all about, and this is all about the clover leaf model structure, clover leaf structure given by clover leaf structure of tRNA described and given by scientist Robert Hole in 1965. Here also presence of adding hyper modified purine. Here in this position H, hyper modified purine is present here as abnormal nitrogen base, also known as T-Psi-CR. Here C is also abnormal base. CAB, methyl cytosine, and this arm is also known as T-Psi-CR. Clear? And what is the function of this arm? With the help of this arm, tRNA combines with larger subunit of ribosome. So this anticodon arm will decide the type of amino acid which will attach to 3' end. And the fourth arm is also called amino acyl synthetase binding site. This enzyme recognizes and activates amino acid so that amino acid is attached to the 3' end of tRNA. In this case, stem and loop both are variable in number. Come to the next and last point, the variable arm. What is this variable arm? Variable, as the name suggests, it has no fixed number of nitrogenous bases. Normally it can contain 4 to 5 nucleotides, but they can contain up to 24 nucleotides. Variable arm, normally it contains 4 to 5 nucleotides, but can contain up to 24 nucleotides. And based on this, tRNA is classified into class 1 tRNA and class 2 tRNA. Class 1 tRNA and class 2 tRNA. In the case of class 1 tRNA, 4 to 5 nucleotides are present, and in the case of class 2 tRNA, 10 to 24 nucleotides are present. Okay, 10 to 24 nucleotides are present. A new class 2 tRNA is discovered more recently, serve as adapter for selenocysteine. And this selenocysteine, as per our knowledge, we know that this is 21st amino acid. Normally there are 20 amino acids, essential amino acids are present, sort of we know, but there is a newly found amino acid is present, which is known as selenocysteine, and a separate tRNA molecule, 2 tRNA molecule is present for this amino acid selenocysteine. tRNAs are thought to be folded into L-shaped 3D structure necessary to fulfill other functions. Each cell or organelle, there are 20 families of tRNA, one per each amino. Iso-accepting tRNA belong to the same family and charged with the same amino acid during protein synthesis. The tRNA are monomeric. It can be said that it is monomeric while arising from one gene, and polymeric along from many gene, arise from many gene. From this structure we have already known that it is already known that there are lots of abnormal bases. So what is the function of abnormal bases? Why while all these lots of abnormal bases are present in the structure of tRNA, what are their functions? Why are those present in a large number in case of tRNA? They are important because they protect tRNA molecule against degradation by RNases. This protection is necessary because this RNA is found floating within the cell. So it's all about of tRNA. My next lecture will be about rRNA.